the sugar molecule will be attached to a phosphate group, that is PO4. It will be attached to a phosphate group. So all these together we call it as a nucleotide. So DNA is deoxyribonucleic acids. What? It contains a deoxyribose sugar which is connected to the nucleic acids. Friedrich Mischer was the first person to say that there is an acidic material that is present inside the nucleus of the living organisms or the eukaryotes, and he called it as the DNA. So that was the first discovery of the DNA. Hello everyone, a warm welcome to this session for class 12 biology. I'm Dr. Divya, biology faculty. So in this session, we are starting with a new chapter, that is chapter 5, molecular basis of inheritance. So in the previous session, we learnt about Mendelian genetics. We learnt about factors or genes. So in this session, where are the genes exactly present? That is in DNA. What is the structure of the DNA? All that we will look into. So to begin with, we need to know about nucleotides. If you remember in your class 11th or in your first PUC, you would have learnt about this in the chapter biomolecules, right? Wherein you have studied the difference between nitrogenous bases, the nucleotides and the nucleosides, right? So what are these nucleotides? So nucleotides are nothing but which are made up of three components. It contains three important components. So one of the component is nitrogenous base. That nitrogenous base, if it is a DNA, what can be the nitrogenous base? It can be adenine, it can be guanine, it can be cytosine and thymine. And if the molecule is a RNA, if it is a RNA, then a nitrogenous bases will be adenine, guanine, cytosine, and will they have thymine? No. The main difference between a RNA and a DNA is DNA, one of the nitrogenous bases, thymine, but in the case of RNA, they do not have thymine. So instead of thymine, they have the nitrogenous base, which is called as uracil. So one of the component of a nucleotide, and among the three components, one of the component is a nitrogenous base, which can be adenine, guanine, cytosine and thymine if it is a DNA, or it can be adenine, guanine, cytosine and uracil if it is a RNA. Now talking about the other component, which is the sugar. This is the second component. So sugar, here also it can be two types of sugar. One is ribose sugar and the other one is deoxyribose sugar. So if it is a DNA, it will have a deoxyribose sugar, wherein in the 2' end of the carbon atom or 2' end of this particular ring, they will not have a hydroxyl group. So that is why it is called as deoxyribose sugar. And the one more, if it is a RNA, they will have a ribose sugar. So again here, these are one of the components. So the second component, if it is a DNA, it is a sugar, right? So the sugar will be deoxyribose if it is DNA, and it will be a ribose sugar if it is a RNA. Both are pentose sugar itself, but the only difference is if it is DNA, it is a deoxyribose sugar, RNA it is ribose sugar. So this is the second component. Next, moving to the third one, that is the sugar molecule will be attached to a phosphate group, that is PO4. It will be attached to a phosphate group. So all these together we call it as a nucleotide. Therefore, be it the genetic material, either DNA or RNA is made up of what? They are made up of nucleotides, wherein they contain three components. One is the sugar, which is, if it is a DNA, it is a deoxyribose sugar, which will be connected to one of the nitrogenous bases, any one, and it will be, on the other end, it will be connected to a phosphate group. And if it is a RNA, the same thing, one of the component will be a sugar, it will be a ribose sugar, one end of the ribose sugar is connected to the nitrogenous base, any one of these, and the other end is connected to the phosphate group. So therefore, the backbone of the genetic material is always made up of phosphate. It is important for MCQ. So this is about the nucleotide. So nucleic acids, they are polymers of nucleotides, right? So what are nucleic acids? They are nothing but those components present in the DNA. So say for example, we have, these are the A, T, G, C. These are the nucleic acids. Altogether, they are polymers of nucleotides. And deoxyribonucleic acid and ribonucleic acid are two types of nucleic acids which are found in living systems, right? So, humans or almost all the animals, that is the eukaryotes, we have DNA in our body. Whereas the prokaryotes, that is the bacteria, or maybe some of the viruses, all those have RNA. So they can have, some of them can have both DNA and RNA, also the genetic material. Especially viruses, they have RNA as the genetic material. So that is those two are the nucleic acids, that is DNA and RNA. What are they made up of? They are made up of nucleotides, which contains nitrogenous bases, sugar and phosphate. So moving further, now we have an idea of what are the nucleic acids made up of. So how many types I told you? Two types, DNA and RNA, right? So in this session, we'll concentrate on DNA. So DNA is the genetic material in all the animals, in plants and we human beings, that is in the eukaryotes. So nucleic acid in that DNA acts as the genetic material in most of the organisms. So whatever our skin color, the texture of our hair, the color of our hair, our eye color, everything in our body, or the biochemical functions, the metabolic processes, all those, everything is controlled by the DNA in our body, and therefore it is the genetic material in most of the organisms. Now coming to RNA. So DNA is deoxyribonucleic acids. What? It contains a deoxyribo sugar which is connected to the nucleic acids, right? And RNA is ribonucleic acid because it is made up of ribo sugar, one of the components. So RNA, though it also acts as a genetic material in some viruses, it mostly functions as messenger, right? So messenger RNA. So during the process of transcription, translation and all that in our body, if the genes have to synthesize proteins and all that, RNA, messenger RNA, that is, we call it as mRNA. These are very important. So what is the importance of mRNA? In the coming sessions, we'll learn about it when we talk about transcription, translation and all that. Okay. So mRNA, we all have this messenger RNA, which is very, very important in our body, for the normal functioning of a body. Next, RNA has additional roles as well. It functions as adapter, it functions as structural and in some cases as catalytic molecule, because any process that has to take place in a body, the nucleotides which are there on, or the nitrogenous bases which are there on the DNA strands has to be read by the messenger RNA. So therefore messenger RNA is very, very important. Though we have DNA which is a genetic material, if messenger RNA is not there, then none of the body functions will happen properly. So it's very, very important. You will understand that in the coming sessions. So this was about the DNA and the RNA. And RNA usually, they are single-stranded, whereas DNA is double-stranded. Can you see? They have two strands. This is one strand, this is the second strand. Therefore, the DNA is double-stranded. And that double strand is always in the form of a helix. That is, it is in the form of a ribbon or a coil. So, this is about RNA and DNA. So, next let's move on to learning about how the DNA was discovered. So, DNA as an acidic substance present in the nucleus was first identified by Friedrich Mischer in the year 1869. So, Friedrich Mischer was the first person to say that there is an acidic material that is present inside the nucleus of the living organisms or the eukaryotes, and he called it as the DNA. So, that was the first discovery of the DNA by Friedrich Mischer. Next, he named it as nuclein. So, therefore, during his discovery, he didn't call it as DNA. He called it as nuclein. However, due to technical limitation in isolating a long polymer, DNA, they are stretches of long polymers. And now you can ask me, ma'am, they're stretches of long polymers. How will they fit into the length of a cell? Our cells are so tiny, right? How do they fit in? That we'll study in the next session. That is also quite very interesting, how they fit inside one small nucleus in a cell. So, we'll learn about that. So, because it was all because of some technological problems or no advancement in science and technology and all that, he was not able to identify it. But he observed it and he said that there is something inside the nucleus and he called it as the nuclein. And it was in 1953. that James Watson and Francis Crick, based on X-ray diffraction technology. They used X-ray diffraction technology, and that data, that is X-ray diffraction data, was given by Maurice Wilkins and Rosalind Franklin. So therefore, with the help of Watson and Crick, with the help of Wilkins and Franklin, they proposed a very simple structure of the DNA, which is called as the double helix model of the DNA. So that is when they told DNA is double stranded and it occurs in the form of a helix. So these are the contributions which have been done by scientists in the discovery of the DNA. So they found out DNA, but how did they understand that DNA is a genetic material? That we'll study in the next coming session. So talking about the DNA, now we know how DNA discovery happened. So we'll talk about the structure of the DNA. So DNA, it is a long polymer of deoxyribonucleotides, right? I told you what are nucleotides. Now when I say deoxyribonucleotide, it is understood. It is made up of deoxyribose sugar, and it contains a phosphate group, and it contains one of the nitrogenous bases or a nucleoside. So the length of DNA is usually defined as or defined as number of nucleotides. So we can say the length. Say for example, number of nucleotides. We have adenine, guanine, cytosine, thymine, again adenine, thymine, cytosine, guanine. By counting the number of nucleotides that are present, they say this is the length of the DNA. So that is how it is measured. And say for example, do you think all the DNAs will have the same length? No, it depends on the organism. Humans have a different length of DNA. Bacteriophages have a different length of DNA. Bacteriophages, I told you, some of the viruses which infect the bacteria, they can have both DNA as well as RNA as the genetic material. So here, for example, a bacteriophage known as lambda into 174 has 5,386 nucleotides. So that is the length of that particular DNA. Bacteriophage lambda has about 48,502 base pairs. That is the number of nitrogenous base or nucleotides present in them. Escherichia coli, which is a bacteria that is present in the intestine of human beings, that also has 4.6. It is 4.6 into 10 to the power 6. That is, it is 10 times 6 times more. So it is 4.6 into 10 to the power 6 number. It is very lengthy number. So that is why they write it as 4.6 into 10 to the power 6 base pair. And haploid content of human DNA. Our human DNA, the length is about 3.3 into 10 to the power 9 base pair. So this is the DNA that is present in humans. This is important. It can come from MCQ. What is the length of the human DNA? It is 3.3 into 10 to the power 9 base pair. So this is about the DNA. So here, as you can see, we have taken the example of virus. This is one form of a virus. Then here we have taken the example of a bacteriophage. So all these have different lengths of DNA. Then we have E. coli, humans. And human genome is the one that is entirely sequenced. So we know what are the different nucleotides present in the body, where exactly the nucleotides are positioned in DNA. Entire human genome sequencing has been done. So we'll learn about that in the last topic of this particular chapter. So moving further, we'll learn about the structure of polynucleotide chain. Again, what do we mean by polynucleotide chain? Poly means many. So they are made up of many nucleotides. So they are made up of many nucleotides, meaning they are made up of many phosphate, sugar and nitrogenous base groups, right? So that is what we will learn here. So talking about the structure, this is a simple polynucleotide chain that is given in the NCERT textbook that we have drawn here. So here, because it is a double helical structure, now here also the same thing will continue, okay? But here, because it is A, it will be T. Because it is T, it will be A, because we know that adenine always base pairs with thymine with a double hydrogen bond, and guanine always base pairs with cytosine with a triple hydrogen bond. Adenine cannot base pair with cytosine, guanine cannot base pair with thymine. So this is the rule of the DNA. This is how they base pair. So here, therefore, it will be C. Here it will be G. because it is a double stranded structure. So here one single strand has been shown here for better understanding. So what did we say? What are we learning about? We are learning about the structure of the nucleotide, right? So structure of the nucleotide. Can you see here? There is a phosphate group. One of the component is a nucleotide is made up of 3 components, right? We have learned it in the first slide. So the second component is a deoxyribose sugar, and the third component is this one, that is a nitrogenous base. Here it is adenine. Next, moving further, they are polynucleotides. They are made up of chains of nucleotides. So that is why they are first, second, third, fourth, like that it keeps on continuing. So if it is a human, how many nitrogenous bases will be there? 3.3 into 10 to the power 9 times. Understood? So like that. We can't write everything, right? So a bit will be shown for better understanding. So here moving further, here also the same thing. Again, it is attached to a phosphate group. This is a deoxyribose, then this is a nitrogenous base. So that is what DNA is made up of. They are made up of chains of nucleotides. And apart from that, on one end, that is, they have the phosphate group is attached to a 5' end, and the hydroxyl group of the deoxyribose is attached to the 3' end. So therefore, it is a 5' phosphate end and a 3' hydroxyl end. So this is how. And next, this one, here it will be 3' hydroxyl end and 5' phosphate. So 3' and 5'. So that is how it will be. So moving further, what are the salient features of the double helix structure of DNA? Now we know what nucleotide are, how a polynucleotide chain is formed, which is nothing but the DNA, what it is made up of. So we'll study what are the salient features of DNA. So it is made up of 2 polynucleotides, as I told you, right? Why 2 polynucleotide chain? Because it is so DNA will be wound like this. It has how many? 1, 2. So therefore, it has 2 nucleotide strands, right? So here, adenine, guanine, cytosine, for example. Then it will be thymine, it will be cytosine, guanine. So 2 nucleotides. So therefore, it is made up of 2 polynucleotides. See here, they have shown 2 polynucleotides. So where the backbone is constituted by a sugar phosphate group. So can you see here, a sugar phosphate? Why are we calling it a sugar phosphate? Because this is a sugar. Right. What is that sugar? It is a deoxyribose sugar, and this is a phosphate, PO4. It's a phosphate group. So therefore, the backbone of DNA, why it's called as the backbone? Because it is towards the outside. Adenine is towards the inside. So we don't call nitrogenous bases as the backbone. We call the phosphate sugar molecule as the backbone. And the base projects inside. That's true, right? The base. This is the base pair, or this is the nitrogenous base. For example, adenine here, it projects towards the inside. So the 2 chains have anti-parallel polarity, right? They have anti-parallel polarity because one chain begins from the 5' end and ends in the 3' end, and the other chain begins in the 3' end and ends in the 5' end, which causes a polarity, making them to twist. So that is about this. So moving further, the bases in 2 strands are paired. Now, if the 2 strands have to be held together in position, there has to be something which holds them. What is that? It is a bond, right? What type of bond? It is a hydrogen bond. Now, if the bond is not there, one strand will be somewhere, the other strand will be somewhere. They will not coil properly and they will not be held in position properly. So, that is why this hydrogen bond that is there, it is very, very important. Now, here, talking about the hydrogen bond. So here, if you can see adenine, so these 2, that is the one of the nucleotide strand that runs from the 5' to the 3' end, and the other nucleotide strand that runs from the 3' to the 5' end are held together at the places where base pairs are there or nitrogenous bases are there by what bond? It's called hydrogen bond. And adenine always base pairs with thymine, and they are held together by 2 hydrogen bonds. As I told you, adenine is always held together by 2 hydrogen bonds. That is why they form a bond now. So it won't get, the strands will not get separated. Similarly, thymine, with 2 hydrogen bonds, base pairs with adenine. Here, guanine, base pairs with cytosine with 3 hydrogen bonds. Same way, cytosine, base pairs with guanine with 3 hydrogen bonds. So this is about how they are held together. And always the purines come opposite to a pyramid. Pyrimidine. Say for example, we have adenine and guanine are the purines. Thymine, cytosine is the pyrimidine, for example. So always they are opposite to one another, right? So if there is a purine here, here it will be a pyrimidine. Here it is a pyrimidine. Here it will be a purine. Again here purine, pyrimidine, pyrimidine, purine. So that is how they are arranged. How they are held together? By hydrogen bonds. So next, moving further, the 2 chains. Now we know how they are held and all that. Now we are talking about the coiling. Why do they coil? So the 2 chains are coiled in a right-handed fashion. That is towards the right hand, not towards the right. Left, towards the right. They are coiled towards the right-handed fashion. And the pitch of the helix, that is between the 2 helix, it has a distance of 3.4 nanometer. It's the distance between the 2. That is, in each turn, base pair, 10 base pairs will be present in each turn. So the pitch between each turn, it is 3.4 nanometer, and there are roughly 10 base pairs. So in each turn, that is, in this turn, how many base pairs are there? 10 base pair. In this turn, if we count, there will be 10 base pair. In this turn, there will be 10 base pair. So for every 10 base pair, there is coiling of the DNA. Understood? No? And the coiling is towards which side? Towards the right-handed side. So that is what it means. Now the distance between the 2 base pair, it is about, not 2 base pair, between the 2 strands, it is about, or 2 helix, it is about 0.34 nanometer. That is this distance between these 2 helix is how much? It is 0.34 nanometer. So this is about the length, the distance and the pitch of the DNA, or at what length, if the coiling has to occur. So where exactly can the coiling happen? Anywhere it wants? No. After every 10 counts of the nitrogenous base, the coiling will occur. So it is about 10 base pair, after every 10 base pair. Moving further, we'll talk about central dogma. Central dogma, being a biology student, central dogma, you shouldn't forget about it throughout your life, because it is very, very, very important. Without the central dogma principle, whatever genetic material we have in our body is also a waste, because the genetic material, the genes, for that to produce into proteins, which performs various, proteins are nothing but chains of amino acids, which performs various functions in our body, which decides how our body needs to function. Everything is controlled by the central dogma principle. So it is very, very important. So the central dogma principle was proposed by Francis Crick, and it states that the genetic information flows from DNA to RNA to protein. It's an important MCQ. From where? The genetic information from DNA, because the genes are present in DNA. From there, it will flow to RNA. Now, which is the RNA I'm talking about? I'm not talking about the viral RNA. I'm talking about the messenger RNA here. You remember, first I told you in the first slide, messenger RNA. From messenger RNA, it will produce the proteins, which are nothing but chains of amino acids. And those proteins are the ones which decides how everything in a body. So the entire functioning of a body is controlled by the genes. Any alteration, any mutation can lead to disorders and diseases. So this is, and in some viruses, the flow of information is reverse direction from RNA to DNA. Okay. In eukaryotes, in all the eukaryotes, the flow of information according to central dogma is from DNA to RNA, and then the proteins will get produced. But in viruses, it is from RNA to DNA. Remember that, it can come from MCQs. So here, first DNA. Now, if DNA information has to flow from the parent cell to daughter cell, can the same DNA strand enter into the daughter cell? No. Then the parent cell will lose its DNA, right? So for that, what they should do? They should make copies of DNA. So that is the first step, where we call it replication, meaning replica or copies of the DNA will be produced. After the copies of the DNA is produced, then the transcription process will take place. So in the transcription process, the mRNA, it will come to transcribe. So what it will do is, say for example, this is the DNA strand ATGC. For this, a messenger RNA strand will get produced. Will it have T? No, because it is a RNA. So instead of thymine, they will have uracil. So uracil, then adenine, then cytosine, then guanine. So this is called as transcription. You learn about that in the coming sessions of this chapter, okay? So here, then the translation will happen. For the translation, ribose will come. It will start reading the strands. So that is why it's called as the translation. That is the translator. And then proteins will get produced. So this is the sequential steps that occurs in central dogma principle. It's very, very, very important one throughout your life if you're a biology student. So this was about this session. So we learnt about the structure of the DNA, the nucleotides. We got to know what nucleotides are, what is the structure of the DNA, what DNA is made up of, how they form a polynucleotide strand, how are they coiled, what is their distance between each coil, everything. And we learnt about the central dogma principle. All that we got to know. So in the next session, because we know about DNA now, we have an idea, we'll learn. I told you, DNA, they are quite lengthy. Say for one human cell inside the nucleus, the DNA is 3.3 into 10 to the power 9 base pair. It is very, very lengthy. It can be lengthier than your height, right? Now, in such a small cell, how can they be coiled? How it will be coiled, we'll learn in the coming session, wherein we'll learn about packaging of DNA and how we got to know that DNA is the genetic material. So, the discovery of DNA and all that was done, but DNA is the genetic material we didn't know so far. How did we come to know? We will study about that in the next session. So, see you in the next session with these two topics. Thank you.